Friends, today we'll study about microbial tuberculosis. This is the second video of microbial tuberculosis. So let's begin. Liquid media are generally used for sensitivity testing, preparation, and antigens and vaccines. Various culture media used for mycobacterial isolation include Lowenstein Jensen medium, that is in short LJ, Petragnani, Dorset egg medium. All the three are egg-based, and Middlebrook, that is 7H10, Middlebrook 7H11 agar-based media. Dubose medium and Middlebrook 7H9 are two commonly used liquid media. Petragnani, Dorset egg medium, and Middlebrook 7H10, Middlebrook 7H11 agar-based media. Dubose medium and Middlebrook 7H9 are two commonly used liquid media. Now let's know about the resistance. Mycobacteria are killed at 60 degrees centigrade in 15 to 20 minutes. They are sensitive to ultraviolet rays and sunlight. Bacilli in cultures may be killed by direct sunlight exposure for 2 hours, but bacilli present in sputum may remain alive for 20 to 30 hours. They are relatively resistant to chemical disinfectants and can survive exposure to 5% phenol, 15% sulfuric acid, 5% oxalic acid, and 4% sodium hydroxide. The bacilli are destroyed by tincture of iodine in 5 minutes and by 80% ethanol in 2 to 10 minutes. 80% ethanol has been recommended as disinfectant for skin and rubber gloves. Bacilli may remain viable in droplets nuclei for cultures remain viable for 6 to 8 months at room temperature. Now let's study about biochemical reactions. Microbacterial species Microbacterial species can be identified by several biochemical tests. Some of the important biochemical tests are now we will discuss. First is niacin test. Niacin is produced as a metabolic byproduct by all mycobacteria, but most species possess an enzyme that converts free niacin to niacin ribonucleotide. M. tuberculosis lacks this enzyme and accumulates niacin in the culture medium. When 10% when 10% cyanogen bromide and 4% aniline in 96% ethanol are added to a suspension of bacterial culture, a canary suspension of bacterial culture, a canary yellow color shows a positive reaction. The human tubercle bacilli give a positive test while bovine type is negative. Positive niacin test also occurs in M. simi and M. cheloni. Now let's know about aryl sulfatase test. Aryl sulfatase is an enzyme formed by certain atypical mycobacteria. The organisms are grown in a medium containing 0.001 m tripotassium phenolphthalein disulfate. If aryl sulfatase enzyme is produced, it liberates free phenolphthalein from tripotassium. Then phenolphthalein disulfate. This can be detected by adding 2N NaOH dropwise to the culture. A pink colored drip develops in a positive reaction. Now let's know about the neutral red test. Virulent strains of tubercle bacilli can bind neutral red in alkaline buffer solution, whereas avirulent stains are unavailable to do so. Positive tests are obtained with M. tuberculosis, M. bovis, M. avium, and M. ulcerans. Now let's study about catalase peroxidase tests. Most atypical mycobacteria are strongly catalase positive and peroxidase negative. In contrast, the tubercle bacilli are peroxidase positive and weakly catalase positive. Tubercle bacilli lose catalase and peroxidase activity when they develop resistance to INH. Catalase negative tubercle bacilli are avirulent for guinea pigs. A mixture of equal volumes of 30% H2O2 hydrogen peroxide and oxygen O 0.2% catechol in distilled water is added to a 5 ml test culture and left for few minutes. Catalase production is indicated by effervescence while browning indicates peroxidase activity. Now let's study about next test is amidase test. A typical mycobacteria can be differentiated by their ability to split amides. The commonly used amides include acetamide, benzamide, carbamide, nicotinamide, and pyrazinamide. A 0.01165 m solution of amide is incubated with the bacillary suspension at 37 degrees centigrade unto this is added 0.1 ml of MnSO4 4H2O 1.0 ml phenol solution and 0.5 ml of hypochlorite solution. The tubes are placed in boiling water bath for 20 minutes. A blue color indicates a positive test. M. tuberculosis produces nicotinamidase and pyrazinamidase. Therefore, splites nicotinamide and pyrazinamide. Now, let's study about nitrate reduction test. This test depends on reduction of nitrate to nitrite by an enzyme nitroreductase. This test is positive with M. tuberculosis and negative with M. bovis. Other mycobacteria which may give positive test are M. consacii, M. fortuitum and M. chiloni. The test organism is suspended in a buffer solution containing nitrate and incubated at 37 degrees centigrade for 2 hours. Then sulfanilamide and N-naphthalene diamide dihydrochloride solution are added. Development of pink or red color indicates positive reaction. Now let's know about the next test is susceptibility test. Susceptibility to pyrazinamide. M. tuberculosis is sensitive to 50 microgram per ml pyrazinamide, but other mycobacteria include M. bovis are resistant. Now let's study about susceptibility to thiophene 2-carboxylic acid hydrazine Z. M. bovis is usually susceptible to 10 microgram per ml of TCH. M. tuberculosis is usually not inhibited by this chemical. However, South Indian strains are susceptible. Now let's study about the twin 80 hydrolysis. Twin 80 hydrolysis. Certain mycobacteria possess an enzyme lipase that splits twin 80 into oleic acid and polyoxyethylated sorbitol, which modifies the optical characteristic of the test solution from a yellow to pink. A pink color indicates hydrolysis of twin 80. M. kansasi and M. gordonii are positive, while M. bovis, M. africanum, M. avium complex and M. Escrofulcasium are negative. M. tuberculosis shows variable results. Now let's study about antigenic structure. Mycobacterial antigens are mainly of two types: cell wall, that is insoluble, and cytoplasmic antigens, that is soluble. Now let's study about cell wall antigens. The cell wall consists of lipids, proteins and polysaccharides. The lipid content accounts for 60% of the cell wall weights. Lipids of the cell wall, particularly the mycolic acid fraction, are responsible for the acid fastness of bacteria and the cellular tissue reactions of the body. The cell wall is made up of four distinct layers. Now, let's study about the some points regarding this. First is peptidoglycan layer, that is murin. Murin, the innermost layer which maintains the shape and rigidity of the cell wall, of the rigidity of the cell. Next point is arabinogalactan layer lies external to the peptidoglycan layer. Next point is mycolic acid layer is the principal constituent of cell wall. It is a dense band on long chain alpha alkyl and beta hydroxy fatty acids attached by ester bonds to the terminal arabinose units of arabinogalactan. Now let's study about mycocytes, that is peptidoglycolipids or phenolic glycolipids, form the outermost layer of the cell wall. The agglutinogen antigens have been identified as the sugar moieties on mycocytes. The cell wall antigens include arabinomannan, arabinogalactan and lipoarabinomannan. Now let's study about the cytoplasmic antigens. Cytoplasmic antigens are protein antigens employed to type the mycobacteria. These include antigen 5, antigen 6, antigen 14, antigen 19, antigen 32, antigen 38 and antigen 60. All are protein in nature except antigen 60 which is a lipopolysaccharide protein complex. Now let's study about mycobacteriophages. Some tubercle bacilli are infected with temperate phages. Many mycobacteria infected with bacteriophage are not truly lysogenic. The phage genome appears as independent plasmid. There is no integration of phage genome into the host genome. This is called pseudo-lysogeny. Tubercle bacilli have been classified into four phage types, that is A, B, C and I. Type 1 is intermediate between A and B. Type A is worldwide in distribution and is the commonest type. Infection due to type 1 is more frequent in India and neighboring countries. Type B mainly occurs in Europe and North America. Type C is rarely seen. Phase 33D can lyse all variants of M. tuberculosis but not BCG. Now let's study about pathogenesis. The infection is commonly acquired by inhalation of infected droplets, coughed or like that, sneezed, coughed or sneezed into the air by a patient with pulmonary tuberculosis. In bovine tuberculosis, infected cows develop lesions in the udder and bacilli are excreted in the milk which can then infect people who drink it raw. In developed countries, pasteurization of milk has virtually eradicated this organism. Tubercle bacilli are engulfed by macrophages, but they survive and multiply in macrophages. These lyse the host cell, infect other macrophages and sometimes disseminate to other parts of the lungs and elsewhere in the body. The cell-mediated immunity CMI plays a major role to interact with these macrophages, whereas humoral immunity appears to be irrelevant. CD4 plus helper T cells secrete interferons gamma and interleukin 2, tumor necrosis factor alpha, and others exerting different biological effects. It may result in protective immunity or delayed type hypersensitivity reaction, that is DTH. The one dependent cytokines activate macrophages to kill intracellular mycobacteria and thus result in protective immunity. The two Th2 cytokines induce delayed type hypersensitivity DTH, tissue destruction and progressive disease. Human tuberculosis is divisible into primary and secondary forms. Now let's study about the primary tuberculosis. Inhaled tubercle bacilli are engulfed by alveolar macrophages in which they replicate to form a lesion called gone focus. It is frequently found in the lower lobe or lower part of the upper lobe. Some bacilli are transported to the hilar lymph nodes. The gone focus together with the enlarged hilar lymph nodes is called primary complex. In case of M. bovis, which enters through mouth, the primary complexes involve the tonsil and cervical lymph nodes or the intestine, often the ileocecal region and mesenteric lymph nodes. Now let's study about secondary tuberculosis. It is caused by reactivation of primary lesion or by exogenous reinfection. Reactivation tuberculosis is likely to occur in immunocompromised individuals. Granulomas of secondary tuberculosis most often occur in the apex of lungs. The necrotic element of the reaction causes tissue destruction and the formation of large area of cessation termed tuberculoma. Presence of caseous necrosis and cavities are two special features of secondary tuberculosis. Cavities may rupture into blood vessels, spreading spreading microbacteria throughout the body. The break into airways, releasing the organisms in aerosols and sputum. Open tuberculosis. Now let's study about the immunity and hypersensitivity. Two immunological responses develop simultaneously in naturally infected host. One is anti-tubercular immunity and other being tuberculin hypersensitivity. Both responses are mediated by T lymphocytes sensitized to the bacterial antigens. Humoral immunity appears to be of no relevance in tuberculosis. Immunity and hypersensitivity are two different manifestations of the same mechanism in tuberculosis. In non-immune hosts, the bacilli are readily phagocytosed and multiply inside the mononuclear cells. This intracellular parasitism is associated with the development of delayed hypersensitivity and of activated macrophages which have increased ability to destroy the ingested bacilli. After first infection, the host acquires some resistance against reinfection. In the immune host, the sensitized T lymphocytes developed during primary infection proliferate and release lymphokines which make the macrophages bactericidal. Delayed hypersensitivity can be induced by infection with virulent or avirulent tubercle bacilli. For demonstrating delayed hypersensitivity, tuberculoprotein, that means tuberculin injection, is commonly employed. Now let's study about Koch phenomenon. The response of a tuberculous animal is reinfection. Tuberculous animal to reinfection was best explained by Robert Koch. When a healthy guinea pig is inoculated subcutaneously with virulent tubercle bacilli, the puncture site heals quickly. There is no immediate visibility reaction. After 10 to 14 days, a nodule appears at the site of injection which ulcerates and the ulcer persists till the animal dies of progressive tuberculosis. The regional lymph nodes are enlarged and caseous. If, on the other hand, virulent tubercle bacilli are injected in a guinea pig, tubercle bacilli 4 to 6 weeks earlier, an indurated lesion appears at the site of injection in a day or two, which undergoes necrosis in another day or so to form a shallow ulcer. This ulcer heals rapidly without involvement of the regional lymph nodes or tissues. This is called Koch phenomenon. Koch phenomenon is a combination of hypersensitivity and immunity and has got three components. First is a local reaction of induration and necrosis. Second is a focal response in which there occurs acute congestion and even hemorrhage around the tuberculosis foci in tissues. Third is a system response of fever that may sometimes be fatal. Now let's know about the tuberculin test. First is principle. It delayed or type 4 hypersensitivity reaction. Now let's study about the reagents. First is old tuberculin, in short OT. It was originally described by Robert Koch. It is a crude preparation of 6 to 8 week culture filtrate of tubercle bacilli concentrated by evaporation on a heated water bath. This crude product may lead to serious complications in some patients. It is now rarely used. Now let's study about purified protein derivative. Insert PPD. A purified preparation of the active tuberculoprotein was prepared by Seibert in 1941. It was prepared by growing M. tuberculosis in a semi-synthetic medium. It is called purified protein derivative. Insert PPD. The dosage of PPD is expressed in tuberculin unit, that is TU. 1 TU is equal to 0.01 ml of OT or 0.00002 mg of PPDS. Now let's know about the method. First is Montauk's test. 0.1 ml of PPD containing 5 IU is injected intradermally into flexor aspect of forearm. A PPD dose of 1 TU is used when extreme hypersensitivity is suspected. Now let's study about the half test. This is done with a multiple puncture apparatus that pricks the skin. A drop of undiluted PPD is spread on the area of skin. The multiple puncture apparatus is pressed against this area of skin. Now let's study about the result. In the Montauk's test, the site of injection is examined after 48 to 72 hours and interpreted as like. Let's know about the interpretation. First is positive test. In a positive reaction, there is induration of 10 mm diameter or more surrounded by erythema at the site of inoculation. Positive test only confirms past infection with tubercle bacilli but does not indicate presence of active stage of the disease. The test is helpful in children under 5 years of for indication of active infection. The test becomes positive 4 to 6 weeks after infection of BCG vaccination. Now let's study about false negative. The test may become negative in some conditions. Let's know about these conditions. First is military tuberculosis. Second is when allergy develop following overwhelming infection of measles, Hodgkin's disease, sarcoidosis, lepromatous leprosy, malnutrition, administration of immunosuppressive agents and corticosteroids. Now, next part is false positive. This is observed in presence of related mycobacteria such as atypical mycobacteria. Now let's study about the uses. To measure prevalence of infection in a community. To diagnose active infection in young children, it is used as an indicator of successful BCG vaccination. Next point is laboratory diagnosis. Bacteriological diagnosis can be established by microscopy, culture examination, or by animal inoculation test. Now let's study about the specimen. Specimen collection depends on the site of involvement. Tuberculosis may involve lungs or sites other than lungs, that means extra pulmonary. Next is let's know about the pulmonary tuberculosis. Sputum is the most common specimen. It is collected in a clean wide-mouthed container. A morning specimen may be collected on 3 consecutive days. If sputum is scanty, if sputum is scanty, a 24-hour specimen may be collected. When sputum is not available, laryngeal swab is or bronchial washing are collected. In children, gastric washings may be examined as they tend to swallow sputum. Now let's know about the meningitis. Cerebrospinal fluid, in short CSF, from tuberculosis meningitis TBM often forms a spider web clot on standing examination of which may be more useful than of fluid. Now let's know about the renal tuberculosis. Three consecutive three consecutive days morning uh, samples of urine are examined. Next point is bone and joint tuberculosis. Here aspirated fluid is needed for experiment for testing. Tissue. From tissue, biopsy of tissue can be done. Next is direct microscopy. Smear is made from the specimen on a new glass slide and stained by Gell's Nelson technique referred by Gell Nelson technique. It is examined under oil immersion lens. The acid-fast bacilli, in short AFB, appear as bright red bacilli against a blue background. To detect bacilli microscopically, there should be at least 50,000 bacilli per ml of sputum. A negative report should not be given till at least 300 fields have been examined. Grading of smears is done according to number of bacilli seen. Now, let's know about purulent part of sputum. Purulent part of sputum is used for making a smear. Microscopy is also performed on the smear, which is prepared after concentration of specimen. In case of urine specimen, care must be taken to urine to exclude commensal M. smegmatis bacillus, which is only acid fast and not alcohol fast, whereas M. tuberculosis is both acid and alcohol fast. If a large number of smears are not if a large number of smears are to be examined, fluorescent microscopy is more convenient. Smears are stained with fluorescent dyes such as auramine O or auramine rhodomine and examined under ultraviolet light. The bacilli appears as bright bacilli against dark background. Demonstration of FB by microscopy provides only presumptive evidence of tuberculosis infection, as saprophytic mycobacteria may present a similar appearance. Now let's study about concentration of specimens. Concentration of a specimen is done to achieve homogenization of the specimen, decontamination, that is to kill other bacteria present in the specimen, concentration, that is to concentrate the bacilli in a small volume without inactivation. Such concentration is used for culture and animal inoculation test besides smear preparation. Several concentration methods are in use. First is Petrov's method. It is a simple and widely used technique. Sputum is mixed with equal volume of 4% sodium hydroxide and is incubated at 37 degrees centigrade with frequent shaking for about 30 minutes. It is then centrifuged at 3000 rpm for 30 minutes. The supernatant fluid is poured off and the deposit is neutralized by adding 8% hydrochloric acid in presence of a drop of phenol red indicator. The deposit is used for smear culture and animal inoculation. Now let's know about other methods. Dilute acid, that is 5% oxalic acid, 3% hydrochloric acid or 6% sulfuric acid. Mucolytic agents such as N-acetyl L-cysteine with sodium hydroxide. In a positive culture, characteristic colonies appear on culture medium. Smear is appeared from isolated colony and stained with Zell-Nelson technique. When acid-fast bacillus is slow growing, non-pigmented and niacin positive uh, it is regarded as M. tuberculosis. Confirmation is done by biochemical reactions. In radiometric methods such as BSc-TEC, the growth may be detected in about a week by using carbon-labeled substrates. Mycobacteria metabolize the carbon-labeled substrates and release actively labeled carbon dioxide. The instrument measures carbon dioxide 14 and reports in terms of growth index. A growth index of greater than 10 or greater than or less than 10, greater than or same to equal to 10 is considered as positive. This method can also differentiate between M. tuberculosis and M. bovis, as M. bovis is susceptible to TCH. Incorporation of TCH in the medium inhibits the growth of M. bovis but not that of M. tuberculosis. Now, let's study about animal inoculation. 0.5 ml of concentrated specimen is inoculated intramuscularly into the thigh of two tuberculin negative healthy guinea pigs. Inoculation by subcutaneous route is avoided as it causes local ulcer which may be infectious. The animals are weighed prior to inoculation and therefore at weekly interval. They are tuberculin tested after 3 to 4 weeks. There is progressive loss of weight and tuberculin test becomes positive in animals that develop tuberculosis. Animal is killed after 6 weeks. Now let's study about the autopsy source. From autopsy, what is the conclusion? There are 4 points. First is caseous lesion at the site of inoculation. First is caseous lesion at the site of inoculation. Second is enlarged caseous inguinal lymph nodes. The infection may spread to other lymph nodes such as lumbar portal, mediastinal and cervical lymph nodes. Tubercles may be seen in spleen, lungs, liver or peritoneum. Kidney are unaffected. The identity of the bacteria is seen is then confirmed by demonstration of acid-fast bacilli from the lesions. Infections with pseudotuberculosis, brucella, salmonella, and several fungi may produce lesions which microscopically simulate tubercles. M. tuberculosis is highly pathogenic to guinea pigs and hamsters and virtually non-pathogenic for rabbits, while M. bovis is highly pathogenic for both rabbits and guinea pigs. Cattle is negative and INH resistant strains, and most of the Asian strains isolated from the South India have low virulence for guinea pigs. Sometimes these organisms produce only enlarged lymph node, and the animals may have to be observed for 12 weeks. Guinea pig inoculation is rarely used nowadays for laboratory diagnosis. Now let's study about serology. Serology includes detection of antimicrobial antibodies in patient serum. Various methods such as enzyme-linked immunosorbent assay, that is ELISA, radioimmunoassay, that is in short ARIA, latex agglutination assay have been employed. Several antigens like BCG, antigens 5 and 6, 64 kDa, antigen 60 and K30 kDa protein have been tried for detection of antibody against them. Diagnostic utility of these antibodies is equivocal. Now let's study about molecular methods. Polymerase chain reaction, in short PCR, is a rapid method in diagnosis of tuberculosis. It is based on DNA amplification and has been used to detect M. tuberculosis directly in clinical specimens. The restriction fragment length polymorphism, in short RFLP, is used to type different strains for epidemiological purposes. The principle of this technique is that restriction endonuclease treatment yield nucleic acid fragments of different lengths, the patterns of which are strain specific. DNA probes have been used to identify mycobacterial species isolated on solid culture media or from broth culture. Now let's study about LCR, ligase chain reaction. Ligase chain reaction and transcription mediated amplification are two molecular methods used for detection of M. tuberculosis. TMA is based on amplification of ribosomal RNA as compared to DNA amplification in PCR. Now let's study about chromatographic methods. The analysis of mycobacterial lipids by chromatographic methods such as thin layer chromatography, gas liquid chromatography, in short GLC, and high performance liquid chromatography has been used to identify mycobacteria. HPLC is very specific and rapid method for identification of species. Now let's study about sensitivity testing. Drug resistant mutants continuously arise at a low rate in any mycobacterial population. With the emergence of multi drug resistance in mycobacteria, it is essential to determine the sensitivity testing. The isolated tubercle bacilli are tested in LJ media after incorporating different concentrations of anti-tubercular drugs in the media before inoculation. There are some techniques for sensitivity testing. First is resistance ratio method. Let's know about resistance ratio method. This is the important method. Bottle of LJ medium incorporating doubling concentration of drugs and are inoculated with a test strain and a known sensitive strain. After 3 weeks of incubation at 37 degrees centigrade, culture media are examined for growth. The medium with the lowest concentration of drugs showing no more than 20 colonies is taken as the minimum inhibitory concentration, in short MIC. The result is expressed as resistance ratio. Now let's study about the interpretation. Resistance ratio of 1 or 2 is equal to sensitive strain. Resistance ratio of 4 equals to double fully resistant strain. Test should be repeated. Resistance ratio of 8 is equal to unequivocally resistant strain. Now let's study about absolute concentration method. The MIC of drug against the test strain only is determined. Non-sensitive sensitive strain is not tested for MIC. Therefore, this method is inferior to resistance ratio method. Now, let's study about the proportion method. In this method, the number of colonies growing from a standard inoculum on a medium containing drug is compared with the number of colonies from same sized inoculum on a medium without drug. When more than 1% of mycobacteria grows in the presence of drug, it is regarded as a resistant strain. Now let's study about radiometric method. BSE-TEC radiometric method is most commonly used for each anti-tuberculosis drug tested. A standardized inoculum is inoculated into the liquid media, one containing drug and other without drug. The liquid medium containing C14 labeled substrate. The rate and amount of carbon 14 carbon dioxide 14 produced in the absence or presence of drug is measured by the special instrument and is then compared. Now let's study about the newer methods. Luciferase reporter mycobacteriophage has been used for susceptibility testing of M. tuberculosis. Only viable mycobacteria can be infected with and replicate this mycobacteriophage. Did tubercle bacilli cannot. The isolate of M. tuberculosis to be tested is grown in the presence and absence of drug and the reporter mycobacteriophage is added. Following infection, a substrate of luciferin is added. If bacteria are visible, the luciferin is broken down and light is emitted, which can be measured. This method is named as chemiluminescence. If the isolate is resistant to drug, light will be emitted, while bacteria susceptible to the drug will not emit any light. The amount of light emitted is directly proportional to the number of viable bacteria. Epsilometer test, E-test, has also been applied for susceptibility testing of M. tuberculosis. Demonstration of mutation in specific genes for different drugs is a useful indicator of drug resistance. Now let's study about treatment. The anti-tubercular drugs include bactericidal agents such as rifampicillin, isoniazid, pyrazinamide, that is Z, streptomycin, and bacteriostatic agents include ethabutyl, that is E, thiacetozone, ethionamide, para-aminosalicylic acid, insert PAS, and cycloserine. Short course regimens of 6 to 7 months are used. A combination of 4 drugs, HRZEE, given 3 times a week during an initial intensive phase for 2 months, followed with only 2 drugs, that is HR, 3 times a week during continuing phase of 4 to 5 months. As resistant strains emerge readily by mutation and selection, combinations of 2 or more drugs are used. If patient is treated with only one anti-tubercular drug. Initially, the bacilli die in large numbers, but soon resistant mutants emerge and multiply unchecked. The bacterial resistance may be primary prior to start of treatment or secondary which emerges during the course of treatment. A serious consequence of unchecked drug resistance has been the emergence of multi-drug resistant tuberculosis, in short MDR-TB. The term multi-drug resistance refers to resistance to rifampicillin, pincin, and isoniazid with or without resistance to one or more other drugs. MDR-TB is a global problem, especially in HIV-infected persons. When first-line drugs are ineffective, second line drugs are to be used. Quinolones, aminoglycosides, para-amino salicylic acid, PAS, ethambutol, thiaacetazone, cycloserine, and capreomycin is being used. The directly observed therapy under supervision is being used to prevent deterioration of resistant problem by ensuring the patient's compliance. When the patient does not show improvement, antibiotic sensitivity test is done to detect resistant strains. Now let's study about prophylaxis. Protection from tuberculosis may be done by public health measures, BCG vaccination, and by chemoprophylaxis. General measures such as nutrition and health education are also important. Now let's study about BCG vaccine. Calmette and Guerin in 1921 prepared an attenuated strain of M. bovis by growing it on potato medium. The strain was attenuated by repeated subcultures every 3 weeks for 239 times in glycerin potato medium over a period of 13 years. When the strain became incapable of producing tuberculosis in the susceptible guinea pig, it was named Bacilli calmette guerin, in short BCG. The species M. bovis was selected for vaccine preparation because of the false assumption that bovine tubercle bacilli were of limited virulence in man. The four widely used BCG strains for vaccine preparation include Pasteur, Copenhagen in 1331 and Glaxo 1077. The strains of the BCG, although derived from M. bovis, differ in several aspects from M. bovis. BCG strains grow well on glycerol containing medium. They are aerobic. They are resistant to cycloserine. Now let's know about the dose and administration. BCG vaccine is available in liquid form and freeze-dried lyophilized form. The freeze-dried form is commonly used. The freeze-dried vaccine supplied by BCG Laboratory, Chennai, is reconstituted by sterile physiological saline to make a final concentration of 0.1 mg in 0.1 ml of the vaccine. After reconstitution, vaccine should be utilized within 3 to 6 hours. Vaccine is given intradermally in a dose of 0.1 ml. BCG vaccine should be given soon after birth, following which it may be administered at any time during the first year of life. A small nodule develops at the site of vaccination 2 to 3 weeks after injection. It increases slowly in size and attains a diameter of 4 to 8 mm after about 5 weeks. It then subsides or breaks into a shallow ulcer which heals spontaneously, leaving a 4 to 8 mm diameter permanent round scare. Such individuals become tuberculin positive after 4 to 6 weeks. Now let's study about protective efficacy. A number of BCG vaccine trials were undertaken and the result varied from 0 to 80%. The immunity has been reported to last for about 10 years. The general opinion at present is that BCG does protect from tuberculosis. Even if disease occurs, it runs a milder course in vaccinated children. It is also believed to prevent serious forms of tuberculosis such as meningeal, skeletal, and miliary form of the disease. BCG vaccine stimulates T lymphocytes, therefore it may offer some protection against leprosy and leukemia. Now let's study about complications. Complications are rare. Occasional complications include local abscess, keloid formation, and indolent ulcer. Then is regional lymphadenitis. Third point is systemic, that is fever, mediastinal adenitis, erythema nodosum. Next point is contraindications. BCG vaccination is contraindicated in patients of AIDS, eczema, pertussis, measles, and patients on steroids. Now, let's study about chemoprophylaxis. It is advocated only in some risk groups. Let's know about this risk groups. Individuals who are in unavoidable contact with a patient with open tuberculosis, example baby born to a mother with the disease. Then is tuberculin positive but radiologically clear unvaccinated children. Next point is adults with the radiological evidence suggestive of inactive disease. Next is these individuals are given isoniazid alone with an assumption that the bacillary load is small and the chance of emergence of a drug resistant mutant is remote. Finally finished, it is I have finished tuberculosis. Any questions regarding this you can ask me in the comment box and subscribe my channel to know more updates. Thank you.